Yeah, go ahead.
Okay, so hi everyone. So now I'm going to give a semi-research talk. It's also about tools for modeling. So in a way, it's new stuff, but it's also helping you illustrate a little bit about how to build some models. So I hope this is useful. But anyway, it's kind of a research talk here. So I'm going to talk about constructing models that are composed of basic units that are connected in some way. And a lot of models can be classified in that way, as you'll see. In some cases, some that may not or initially be obvious that they are units connected. So hopefully I'll talk about them. And that means that I'm also discussing a new tool that I created called SB Modeler, which stands for Systems Biology Model Replicator. That R stands for replicator. And I'll talk much more about it. Okay, so I'm going to talk about what's the motivation for this and a little bit background. And then I'll also describe how you can make COPASI programs in Python. So Python programs that use COPASI in the backend. And then I'll describe the SB Modeler software and some examples. Hopefully, if I have time, I can show you some nice examples. Okay, so motivation for this. So why do we want to make models, complex models based on a number of units that are connected? The motivation comes from essentially thinking about cells living in the real world, where they're normally never a single cell alone. Normally, you have communities of cells and a certain environment. And for example, you can have microbial communities. This is very common, right? So you're growing a culture of bacterium. By the way, this one here is the bacterium that was mentioned in one of the lectures yesterday. But so you can have, so here you have a number of cells, and all these cells are more or less the same. You can consider them, or at least for modeling purposes, that they're all similar. And so you could have a model that represents a single cell and then replicate that model to have a model that represents the entire culture. Same thing happens with tissues. In tissues, let's say animal or plant tissues, the cells are joined together somehow, and they also communicate. They share some of their contents and transport certain things between them, etc. Or you also can have organoids. These are kind of little organs created in the lab that try to replicate certain particular tissues and organs, particularly in animals. Those are also a number of cells connected with potentially with an environment around them. That's an important point that I also should highlight. In all these three cases, not only can the cells communicate directly between them, for example, here between cells in the tissue, side by side, say with gap junctions, but they can also secrete molecules to either the medium around them or maybe the certain transported liquids, such as the plasma of the blood plasma. And then other cells can take up the things that one cell secreted, the other cell may be able to take up. So there's some interactions that may not be directly between cells, but sometimes through the medium. So we want to be able to create models of these things in an easy way. There's a fourth option here that may not be so obvious now, but hopefully I'll have time to explain it, which is you can also have models that are essentially networks of little motifs that are connected in some way. So for example, gene regulatory networks are motifs of gene expression of a specific gene connected to another gene by induction or repression through proteins. Those things can also be seen as basic units that are replicated. Okay, so that's the motivation for this work. And now I will take a little detour to tell you about Basico. So I mentioned this yesterday in the COPASI description. This is a Python package that allows you to run COPASI from Python scripts. And essentially, it's an interface. It gives you a number of functions that you can call, and those functions will basically do something with COPASI on the backend. So essentially, the code is still being run in the C++ compiled code of COPASI, But rather than being driven by the front end, which is the normal way in which Copasi works, rather than being driven by the GUI, the graphical user interface, now it's being driven by the statements you write in your Python program. So this is useful to write just basic scripts, like some kind of workflow, or you can write Jupyter notebooks. And essentially, you could even replicate what you do with the graphical user interface, but now in a way that it's programmed so that that program can be replicated and run several times. So you could think of them as a macro of what you would do in the GUI. Basico can also be used through Google Colab and I think some other cloud type computing environments. It's a project that is hosted on GitHub. It was built by Frank Bergman, a colleague of ours, a collaborator from Germany. It's basic package now. You can install it in Python using PyPy, so pip install, simple commands. Documentation exists in read the docs. And Frank has now published a paper very short paper that describes the software and is a good reference to cite. So I'm just going to show you, basically this is the read the docs interface with the manual. So you have everything, so examples, etc. How do you work with various things, and gives you examples here. It also has the full API, what are all the functions, etc. So it's very useful. That's the paper that was published in Journal of Open Source Software. And if you don't know how to get to this, you can go to the main Copasi website, and here under projects, you have Basico is actually the first entry. There is also a similar package for the R language, which I mentioned yesterday. I'm not going to talk about it, but just so you know, you could also access it through this. And as you can see, you can also access SB Modeler from this same link. Okay, so just some quick examples of what looks like to use Basico. So in this first one, concentrate on this column on the left. This is a very simple script that just loads a model. So first you start your program importing the Basico package. Of course, you would have had to install it before, which is done with pip, as I mentioned. Then you just load a model. In this case, we're loading a simple model from the current folder. And then you just run a time course. That's the function that says, okay, run the time course. That time course is programmed in this model that you obviously saved before with Copasi, or someone has saved before in Copasi. So this will run the time course that is programmed there, and the results go into a data structure, pandas data frame. And so because you're using Python, so if you have pandas and matplotlib, you could just quickly create a plot just by doing dfplot in this case. So you created this structure, and then you just plot, and it plots the results of the time course. So the results actually get stored in Python in a current data structure. That's a very simple example. This is the equivalent of loading that file in Copasi, going to the time course task, and press the run button, and having defined the plot to then see the outcome. On the right side, it's a little bit more complicated just because it has more operations, but it is now doing metabolic control analysis. So we have the beginning is the same. We load the model, we import Basico, we load a model, and then we just tell it to get the elasticities. That will show you immediately, will tell us, shows the matrix of elasticities, which is one of the coefficients in metabolic control analysis. Then if you want to get the control coefficients, in this case, the concentration control coefficients, again, it's just running this simple command, which will return a data frame with all the concentration control coefficients. And then finally, if you want to get the flux control coefficients, it's just another simple command, and you get a data frame that has the flux control coefficients. These are just two short examples. But what this allows you to do, as you can see, is then you can create much more complex programs, which I will show you in a moment. That's what SB Modeler is. It's basically a program written in Basico. So essentially, it's using Copasi to do a number of operations that are more complex than what you would normally be able to do with just a straight Copasi interface. Now, Basico supports a bunch of operations. Frank Bergman is actually still building it. So whenever there's a need for some functionality that doesn't exist yet, something you can do in Copasi that you cannot do in Basico yet, he normally programs that in quick time, like a week or so, and he releases a new version already with a new functionality. But these are the ones that are currently supported of functions that Copasi can also do. So model editing, time course and steady states, parameter scans, sensitivity analysis, parameter estimation, optimization, etc. And then it also provides some more complex functions that you cannot readily do in Copasi in the GUI. For example, profile likelihood. I mean, you can do it in the GUI, but you have to program a complex set of parameter scans and plots and parameter estimation. You can have to link all of those. This will run everything in Basico in the background. Also, it now has an option for doing model selection that is using Kaike's information principle to select different models. It can load PTAB is a format for parameter estimation that was proposed a number of years ago, and Copasi cannot read PTAB files directly, but through Basico, Basico reads the PTAB and creates the equivalent Copasi file. So in Basico, one could write very quickly a converter for PTAB to Copasi. And also it can get models directly from biomodels and JWS. These are two different model databases. Copasi can actually also do the same if you know the URLs. So those are the things that Basico does. Hopefully, if some of you are interested in writing programmatically controlling Copasi this way, then this here is the hint for you, what you can do. And during the rest of the sessions, I can talk about this later on. Okay, so back to my building large models with small units. I had already done something like this more than 20 years ago in a program called MEG, which was for my previous software called Gepasi, which is essentially the precursor of Copasi. And MEG was already destined to do that. So you would take a Copasi simple model, and it would then create a more complex model that was a repeat of that simple model. And it allowed you to do basically cell cultures or tissues. So this here are showing you what we call cell in the soup. I think it's called cells in the soup, which is basically a number of units that only communicate with the medium and also creates a compartment for the medium. It can create an array of units, rectangular array of units, which are now basically cells which can communicate with each other through some kind of gap junctions. Or then the same thing, but also embedded in a medium. These were the three topologies that it would allow. And then you could select which are the species that can be transported between units, and maybe even making some parameters randomly distributed so they could be some parameters being different in each cell with some properties here. And then this would generate a new Gepasi file, that now has a large number of units. It's a much larger model that rather than having just one cell now has maybe hundreds of cells. So this was already in existence. And in fact, in Copasi, in the actual GUI, we did create a basic structure that can also do the same, called arrays of compartments, which you can get from the tool menu. So if you do that in your model, you can define certain compartments that to be replicated and make it as a linear chain or a rectangular array. So a little bit of the functionality is there. However, it doesn't work with everything. It doesn't replicate, say, your parameters or your events or other things that your model may have that are not just the reactions and species. It would not replicate those. And so even though it can do some of this, it doesn't do everything that I really wanted to do. So that's part of the motivation. And then the other motivation was to create models that link. So if you have a model of a neuron, of the electrophysiological model of a neuron, we would like to be able to connect them. So have create models of neural networks, spiking neural networks, they're usually called, where the neurons again, communicate with each other through synapses, either through so-called gap junction synapses or electrical synapses, sometimes they're called, or chemical synapses, which are those that use neurotransmitters. So we wanted to do things like that. So having neurons, model of a certain neuron, and then being able to connect several of those neurons together and make a bigger network of neurons. And then finally, again, something that I had done in the past as well, which is create arbitrary gene networks. This is something that I also created about also 20 years ago also, whereas this little program would allow us to create arbitrary gene networks, like, for example, these two examples here, where each unit is a gene that is connected to another gene by induction or repression. So we would have kinetic functions that replicate induction or repression, depending on the values of the parameters. And then each unit is a gene that can be activated by a arbitrary number of other genes, activated or repressed. Okay, so then I decided that it was time to make a new version of that old program I had and make it for Copasi. And that is essentially what I'm going to describe in the rest of the talk. It's called SB Modeler, as I mentioned. Its initial name used to be called Model Replicator, and then I just fused it into a single name. It's also on GitHub. It's basically, I showed you already how you can get there through the Copasi main page. You can install it with pip as well. It's already available in PyPy, even though this is not the definitive function, the definitive version. You can already import. And of course, it's written in Python using Basico. As you can see, it imports Basico. And it also is a large program, so it has around 2,300 lines of code. So it's not exactly a short script. It's a complicated piece of software because it does a lot of different things. It is a command line program, so you actually normally call it from the command line. So this is the help file that you would see if you typed sbmodeler -h. You get a list of all the options that it has. Essentially, you use it on the command line and you call it with a model that is your base unit. So you have to already have a model, which is a model of your base units. Let's say a model of your neuron or a model of your cell or a model of your gene expression. And then you tell SB Modeler how you want to create the network. And that's what the rest of the talk I'll describe, but essentially it can be just single units embedded in a medium, like I mentioned, cells in a culture. It could be single lines of cells. It could be 2D rectangular arrays of cells. It can be 3D cubic arrays of cells, cuboids. And it can be some arbitrary network connections, which we'll talk about. Okay, so this is just to say that it is currently a command line program. You basically run this in the command line and you go from your single model to a model that is much larger. Then you can load that into Copasi and do whatever you want with it. So what are the topologies I mentioned? So you can have, you can go from a unit here, which in this example, it's a unit of a single cell, in this case, that has only 3 reactions. And you can make a model of 3 cells that are independent, right? So this would be the call. This is what we call 1D, one-dimensional. In this case, they're not even connected. They're just embedded in a medium. Or you can make a rectangular array. Then you tell it the number of rows and columns. So in this case, it makes a rectangular array of cells. And you can also tell it that some species, in this case, X, would be connected between the neighboring units. And SB Modeler would create a model that has all of this. Or you can do it in a 3-dimensional way. In this case, it would be 3 rows, 2 columns, and 2 lines, I think we call them, or the 3rd dimension. But as I mentioned, you can also just create an arbitrary network of connections between the units. So you go from this single unit, and if you give it a special network file, which I'll talk about later, more detail, you can end up with this network where each cell is connected to a number of others. And it doesn't have, in this case, the network, in fact, has a little subnetwork that is not connected to the rest. And it has here some arbitrary network connections. So these are all the topologies that are allowed. Now, for the arbitrary topologies, we use a file format called DOT language from a package called GraphViz. I'm not going to describe the details, but there are ways of describing how units are connected using this language, and that's your input if you want to make an arbitrary network rather than just a regular grid of cells. And these are examples of those network files. They're very simple. So for example, this one here just says that there are two units, 1 and 2, and they're connected. And so that's the outcome. Or for example, here you have a circle. Oops. Here you have a circle where 1 is connected to 2, 2 is to 3, and 3 back to 1. And in this case, the connections are irreversible. So there's transport from 3 to 1, from 1 to 2, and 2 to 3. You can make like a little feed-forward network. Again, these are just very examples showing you that it's very simple to specify the topology. Or you can have a complex one like this one here, which has all of these connections. And it has, this network allows you to also add attributes. SB modeler will read these things, but not do anything with them. But so it can show you that you can use a proper dot graph that you can use to display your network. But SB modeler will only read this part in yellow, which is where the information about the network is stored. Okay, so I mentioned that we can also have cells embedded in the medium. So this is an option that you can add to SB modeler, which is just simply -- add medium. And when you do that, you basically allow not only transport between neighboring units, but you allow transport between units and the medium. So in this case, X is can go from this cell to that cell, but it can also go to the medium. And through the medium, it could go back into that cell. You can also have an option where you ignore compartments. So in the previous case, we replicated the entire cell. So everything that one model has, all the compartments, if this had, like, say, mitochondria and peroxisomes and so on, then each of these would have mitochondria and peroxisomes, just like the base unit. But if you ignore compartments, then what it does is that it creates the new units, but it doesn't put any limits around them. So they're all contained inside a single compartment. This will be useful for the gene networks, which you'll see later on. But you can use it in other contexts. So you can do this, you can add this option, ignore compartments, in any of the topologies. Okay, so that's how the networks are, the units are connected, but then what kind of connections do we allow? So the connections are going to be obviously transport. It's the most basic one. So there are species that are being transported between cells. So that's one thing. So that's basically transport either through mass action kinetics or through Hill kinetics. And of course, Michaelis-Menten is a subset of Hill. So Michaelis-Menten and Hill types. You can also couple ODEs through a diffusive term. This is if you have explicit ODEs. They may be, for example, for electrophysiology. So if you have an ODE that expresses the membrane potential, then that ODE can be connected from one unit to the next unit by a diffusive coupling term. And that's very similar to transport. That allows you to do things such as resistance. So if you have an electrical synapse, you can have basically equivalent of a resistance connecting the two membrane to the two potentials. And you can also add chemical synapses. So in this case, the units, this is very specific for neuron models. Then the synaptic terms essentially create a unidirectional flow that includes a little model of the release of neurotransmitter and the capture of neurotransmitter on the postsynaptic neuron. I'll mention this, I'll describe this in a little bit more detail in a moment. And then the final coupling methodology is coupling units by regulation of synthesis. So in this case, rather than being a molecule that is transported from unit A to unit B, what we say is that there's a molecule in unit A that regulates the synthesis of equivalent molecule in unit B. And this is how we're going to allow us to do gene regulatory networks. So the coupling here is essentially activation or inhibition of some reaction rather than transport. So these are the methods that are allowed, the mechanisms of connecting the different units. So, just to give you, this just gives more detail about each one of them. I'm not going to talk about this in much, I won't spend much time with each slide, but just to show you that when you do transport or diffusive coupling, these are the kinetic functions that couple the two units. And you could see they're very similar. The diffusive coupling is very similar to what happens as mass action transport, is the same, essentially the same kind of equations. And you can have them as reversible or irreversible. The coupling by chemical synapses is a little bit more complicated. So essentially, we're replicating this process that happens when a neuron secretes, or actually could be a molecule that is a cell that is not a neuron, as long as it secretes something that a neuron on the downstream has receptors for that cause an electrophysiological response. This could still also can still also be done. But essentially, you have in cell one, you have something that secretes a substance, which then is diffuses across this little synaptic cleft, and that is hits a receptor on the postsynaptic neuron, which then causes a membrane potential on that neuron. And that is essentially, we follow the work that was described in this paper back in 1994 that allows us to create a very simple model to do this that still more or less follows the biophysics of this process. Now, what this does is that for each synapse, that is each connection between two cells, not per unit, but per connection between cells, it creates an extra differential equation in the model. So if you had a model that had, I don't know, 3 differential equations to represent one neuron, now if you do connection between 2 neurons, then that's, of course, you need 3 equations for neuron 1, another 3 for neuron 2. In this case, if it's a chemical synapse, we're going to have to add 1 extra differential equation for the synapse itself. So that equation describes the amount of the proportion of bound receptor here on the postsynaptic neuron. But it depends on properties of the postsynaptic neuron and the presynaptic neuron, because it includes, summarizes the process of release of the neurotransmitter as well. So that is described by this equation, and it has a number of parameters that have to do with how fast certain processes happen and some of the resting potentials of the process. And I don't have time to describe all the details in all the properties in detail, but if you're interested, then we can talk offline about this. And so that once this is created, then the postsynaptic neuron gets added an extra term in their potential, in their membrane potential equation, and that's how the networks get built. Now, I showed the example with 2 neurons, but you can have an arbitrary large network, and it builds all of those. Of course, it adds 1 ODE per each of the synapses. So this can very quickly get very large if you have a lot of connections between your neurons. And then the coupling by regulated synthesis, this is for gene networks, remember again. Now we look at the unit that is receiving the inputs from other units. That unit basically has a species that is now regulated by other species equivalent to it, but in the other units. And so we have to add a synthesis reaction. So every time there is a connection between 2 units, or in fact, every time there is that one unit receives input from any number of other units, we add 1 new reaction in the receiving unit. So this is the receiving unit here. Let's call it a certain gene. And it's activated by or inhibited by 3 genes, we have to add one reaction in this unit for gene 1, and that reaction is a synthesis reaction that is now regulated by the other 3 genes. And the regulation occurs according to this kinetic type, which is basically a multiplication of terms for each one of the controlling genes, the effector genes, the inducers or repressors. And each one of them has this equation, and depending on the value of this A parameter, you can have it to be activatory, or you can have it to be repressing. So for negative values of A can go from -1 to +1. And if it is -1, it's fully inhibitory. If it's +1, it's fully activatory. And the activation goes according to a Hill function that then can be tuned using this parameter H here. So 2 parameters, A and H. A tells you whether it's inhibition, activation or inhibition, and H tells you how steep is this sigmoid curve going to be. That's basically, and of course, if the receiving gene has 10 different activators, then there will be 10 terms like this multiplied in a row. And then there's also an extra parameter, which is the basal rate. So that's this value here multiplies by the basal rate. So that's the kinetics of the coupling by regulation. Then, if you remember, I mentioned my original program from 20 years ago would also allow you to have some parameters be distributed in some kind of distributions. We also allow that to happen. Parameters can be varied according to either a uniform or a normal distribution. And so you can give it parameters, you can give it the variability that you allow for certain parameters, so they can make models to be, the units not to be exactly to be replicas, but they are slightly different. So you can replicate, for example, what happens when a cell divides in 2 cells, in 2 daughter cells, and the 2 daughter cells don't have exactly the same initial condition because you got more material in one than the other. So there's already some stochasticity in this aspect. We can add that in basically on the parameters of the units. So here are some examples of basically calling SB modeler to make something and the examples. So the first one here, it's very simple. It basically says, okay, I'm calling SB modeler, and I'm going to use this binding KA model. This is the original base model. That's the model that describes a single unit. And now we tell it we want 100 units. And we want parameter KA in this model to be distributed with a normal distribution with 20% variability. That's basically what this says. So the output model, which, because we didn't name it explicitly, is going to have a name constructed based on the base model. That model will have 100 units, and those units will be the same, except parameter Ka will be distributed according to a normal distribution. And because in this case, it's very simple, we didn't tell it to transport anything, so these units are all going to be completely independent. They're just basically 100 copies of the original model, but with slightly different variations on that parameter. The second case, it's very similar to the first, except that rather than adding parameter variability, we're telling it that there's going to be a diffusion. There's going to be a molecule diffusing between units. So we started with a model called calcium spiking, and we said, okay, make this unit, let's make an array of 2 by 2 units, so 4 units. And because it's an array, they're going to be connected, the neighbors are going to be connected. And we say, oh, and we're going to connect this species here called calcium-site. That's a species that exists in the base model. And that's going to be transported basically by diffusive constant. And we also give it a value for that constant of the value of 0.1. Otherwise, it would get a default number. I'm not going to go through every single example. You can see that you get more options. The text below underneath describes it, everything. The last one is basically creating the repressilator model, and it creates a topology, a feed-forward network of repressilator cells, essentially. This one here represents a network of Hindmarsh-Rose type neurons connected through chemical synapses, where the variable X represents the membrane potential, variable X in the Hindmarsh-Rose model. Okay, so these are examples. You can look at them later on. I'm going to describe some use cases I've to illustrate this. So the first one comes from a paper by Janne Wolf. In fact, I used two different papers from Janne Wolf that have worked on these things. So in this paper, they picked a type of model that had been described before by Selkov. I saw a question. I will answer that question in a moment. So a question on the chat. So in this case, we're making a model that has oscillations, and that model we're going to make, rather than just having one unit, we're going to put two units, and we're going to let them communicate through a medium. So one of the species diffuses to the medium, and then it can go from one cell to the other and vice versa. It's completely reversible. And these cells normally have oscillations. And so we're going to see what happens when we connect them. And we're going to connect them basically. So we tell SB Modeler the base Kopasi model, which is here. We tell it that the new model has 2 units of that. So it's going to be the 2 units. We tell it that we're going to transport the molecule called Y. And so this creates a transport. The rate constant for the transport value is here, 3.2. We say that we need a medium compartment, so it adds a medium. And the volume on the medium is 10. And there's a parameter X, capital X, that's the initial condition of X, the initial concentration of X species here. It's going to be distributed uniformly. And the initial concentration of Y is also going to be distributed uniformly. So the 2 cells don't have the same initial conditions. They have the same parameters, but they have different initial conditions. And then we want the output of this to go to a file called case1.cps. So it produces a new Kopasi file that has this whole ensemble. So that basically, doing that, we replicate the results from that paper. That paper also does the same thing, but now with 5 cells rather than 2. So, and that is the same thing. Now it's very simple. You see almost the same line, except then in this case you have 5. Now in their model, they had the different initial thing, so I had to actually use a slightly different, slightly modified version of the base model. But apart from that, everything is very similar, except that this medium has a larger volume, so this is to basically just match the paper. So we can build those models very easily. Whereas in their case, they coded all the differential equations of this 5 unit model. So then it has 10 differential equations. They had to code them all by hand. Whereas here, we just give it the base file and the larger one gets made automatically. And then they have also another variation with 3 cells. That's also the same thing. So that was one example. The next example, it's also, again, using communicating cells, and that's the base model. In this case, it's a model of glycolysis without a catalysis. And what they're going to do is they're going to be doing a 49 by 49 square array of cells, which are oscillating, and they are embedded in a medium. And the media from each of these 49 squares communicates. So now it's not really the cells that communicate, but it's the medium around each one of them. And so there is diffusion between the mediums next to each other. And it's a 49 by 49 square array. So this is a large model. If you think about it, 49 by 49, it's already a fairly considerable number. This is the command line to create the model. It basically says, take that model as the base, have transport by this species X external, give it a rate constant for the exchange of 5, make some parameter, this parameter alpha of the model, give it some variability with a uniform distribution, and then output everything into this file. So it produces a very large file, and I've rendered here the results in this depiction. So this is the domain 49 by 49. So there's 49 squares on each side. And then this is a snapshot at a certain timing point. So in this case, you have every single one of these spots here is this model, and they allow the species X external, this one, to be transported between each square. So that, if you think about it, that's equivalent, almost equivalent to diffusion. If the squares are small enough, that would be exactly the same as diffusion, as Laszlo mentioned. But it is not. It is just transport, mass action transport. And because these cells have oscillations, then what you see is that you have a number of cells here oscillating. Now they're not in phase, and that's because we gave it some slight variation on these parameters alpha. So the oscillations are slightly different amplitude and slightly different frequency. So in this circle here of cells, you have a lot of oscillations that are heterogeneous because of the heterogeneity that we gave it. And then on the outside, there's there is a certain level of synchronization outside of this kind of circle. And this, by the way, is the results that this paper also describes. If you look the paper up, you can also look at that. I'm not going to talk about this here. This is a more complicated experiment, but you can read it in the paper if you so desire. I'm going to talk about now the neural networks. So here we're going to use models that replicate a neuron. So for example, Hodgkin-Huxley model or some variation of the Hodgkin-Huxley model. And we're going to connect them in either simple cases like these here, or in a bit more complicated motifs. These models, these electrophysiological models have equations like that. I'm putting here some examples. We're going to use that approach of creating the synapses that I mentioned before. So in this example here, we're going to make some simple motifs of networks. So in this first case, we have two neurons, one neuron activating another one through a chemical synapse. And the first neuron gets some current input from the outside. It's actually a stochastic current input. And so the outcome is this, and neuron, so that's in yellow, it's what we use to create this model with SP Modeler. Of course, the model here, the base model, RSA neuron, this is a model that includes those differential equations that describe this so-called RSA neuron. And basically, we give it a network. The network just says that it's just one activating two. And we tell it that in this RSA neuron, the variable that is the membrane potential, it's called capital V. And then we create a synapse with a conductivity of 0.08. And this is the outcome. You have neuron 1 is the one that's being given random current inputs. And then neuron 2, at certain times when there is a spike on neuron 1, the spike actually also activates neuron 2. So you see neuron 2 is a slave of neuron 1. Every time neuron 1 triggers, neuron 2 is triggered. But of course, there's little current inputs that happen on neuron 1 that don't cause an action potential. They never get passed on to the downstream, all right? So this neuron 2 is kind of showing you a filtered version of the current inputs. Second neuron is the same thing, but neuron 2 also gives feedback to neuron 1. So this is a full positive feedback network here. And that means that once one spike gets down to neuron 2, the neuron 2 keeps activating. So there's now a train of spikes that eventually die out. Then when the inputs here accumulate for neuron 1 and it spikes again, then there's another train and then another train. So these are, that's the behavior. This, by the way, these results are all simulated with Copasi. And then the final one is a feed-forward, so-called incoherent feed-forward network, where one neuron activates the downstream one and activates the middle one, but then the middle one represses the downstream one. So neuron 3 gets the signal from 2 in 2 different ways. Gets a positive signal directly from 2, and then it gets the same signal, but as a repressive current, repressive potential through neuron 1. Of course, they have different timings as well. So we're showing here neuron 1 and neuron 3. And what happens is that if there is a spike in neuron 1, then it causes a spike in neuron 3. But if there is a second spike in neuron 2, if there is a second spike in neuron 2, shortly after the first, then neuron 3 doesn't get that. So neuron 3 basically, see this one, this 2, it doesn't show. But if there was long enough here between spikes in the first one, then the third will respond to that. But essentially, it cuts out spikes that happen too close in time. So the last one is only showing you when there's a single spike rather than showing you like a train of them. So these are 3 examples, very simple. They all use the same base unit. They're just connecting it in different ways and with different characteristics. The final one, I'm not going to have time to describe too much detail about it, but in this is a replicating a structure that happens in certain animals where they have 2 sides and they have neurons actuating, controlling motor neurons on the 2 different sides. So this happens in worms, for example, or it can happen even in larger animals where if they have this kind of weaving behavior, it's created by activating the muscles on one side of the body alternately with the other side of the body. So the muscles on one side contract when the muscles on the other side are relaxed and vice versa. And that's achieved with a neural network like this, where you have basically certain neurons, this in red, 1 and 4, that are active, that have activatory synapses. And then the neurons in blue, number 2 and number 5, that have inhibitory synapses to all the others. They're connected, you see they're connected to all the others. Follows this structure. This is the vector, the network representation, but this is what their paper have. And it's, again, very simple command for SB modeler. You have the base unit. You tell it just that, well, if you're going to follow this network file that describes how the connections are. And here is the network file, has all the connections. As you can see, all these lines exist 1 to 1. So it has a self-activatory connection. 1 to 2, 1 activates 2. 4 to 4, 4 is also the same thing. Then, for example, 2 to 4, so 2 inhibits 4 or connects to 4. The inhibition or activation is then tuning the values of the synapse potentials. So essentially, you replicate this, and this is what happens in the model. So now you have basically these motor neuron. Well, the activation of the motor neurons. Basically, this is 1 on one side. 3, I think, is this one. No, this one is. I think 3 is the one in green, and 6 is the one in kind of orangey color. So what you see is that there's a train of spikes on one side, and then a train followed by a train of spikes on the other side, and then followed again by 1. So they're basically the 2 motor neurons, these 3 and 6, are basically spiking at alternate times, and that's the consequence of this network here. So I'm getting very close to my end. The very, very last one is really just the gene regulatory network. Here I replicated this paper from Elovitz and Leibler. It's a very classic paper on this transcriptional network that allows oscillations to happen. This is the actual construct that they created. And this is the gene network. There's a gene 1 that activates gene 2, then gene 2 activates gene 3, and then gene 3 back to gene 1. They're actually not activations, they're all inhibitions. So you see here, these are the actual 3 genes that are inhibits lambda CL. Lambda CL inhibits LAC-L, and LAC-L inhibits TET-R. And this causes an oscillation eventually. So we create this network by getting a base file that just tells Copasi to have a gene that is being transcribed, and the product of transcription, the mRNA, then it also has some decay. That's all this network, this file has. And then we tell it, okay, make 3 units of that, so three genes. We don't want any compartments around them, so we have to add the ignore compartments, because these are all still in the same cell. And give it a network that is this 3-circle network, which basically is this file that tells us that unit 1 connects to 2, 2 to 3, and 3 back to 1. And these are the parameter values of the, which are also described down here in this table, the parameter values of the connections between them, and the fact that they're also distributed according to certain uniform distributions. And this eventually creates a model file that has the repressilator created directly from this. Of course, you could make much more complex gene networks. All it takes is to make a more complex network diagram. That's all there is to it. The rest of the command would be very similar. Okay, so then to wrap up, this is still not published. I have not written a paper about this tool, but the tool is available. You can already install it. You can get it from GitHub, or you can install it using pip with PyPy. I want to maybe potentially make a GUI front end for this tool. Perhaps that's not necessarily going to happen, but it's a possibility. I also want to include in these gene networks the proteins, because at the moment it's making the one mRNA directly affect the rate of a gene, which in reality it's the protein that does that. So I would like to make this include also proteins. And then there's a number of small things that could be added, like adding noise for all parameters. So an easy way to describe, make all parameters be noisy. Adding KMs to this regulatory synthesis rate law, which it doesn't have, etc. And that's basically it. So I think I'm spot on time right now. I can open the floor for questions. I know there are a few there, so let me see if I can answer some of these questions.
Yeah, Stefan answered all of them.
Okay. Yeah, so the question about spatial dynamics, yes, it's true. There are no spatial dynamics. But if you look at this case here, if you, in the limit where these units are very small and you have many more of them, that's actually how you simulate 3-dimensional reaction diffusion, basically. And so that's actually this would be what I would say, call a poor man's diffusion here. But note that in this case, it's only one species that is basically being transported by mass action between neighboring media. Now, these cells here are medium, right? It's the big medium. Each one contains a single unit cell in them. So it's like the cells are all scattered and they have some medium around them. They're not really even close to each other. Do all the cells need to be similar? Well, yes. In order to have SB modeler work the way it works, we have to give it just a unit. But I can go around that here in this case. So here we have really different units, right? So these cells are inhibitory. These cells form inhibitory synapses downstream, whereas these cells form activatory synapses downstream. So they were different kinds of neurons, right? So they were neurons with different kinds of neurotransmitters. And essentially, you make the model this way. You have some base unit that you can then go change parameters. So you make the model this way. You get the new model that has all the neurons in there. And then you can just go in load them into Copasi, and then just change by hand the parameters that you want to make it so that you make unit 1 and 4 activatory and unit 2 and 5 inhibitory. So that's the trick of making different cells, but they have to be kind of subsets of the initial base unit, right? Because otherwise it wouldn't work out. If there's any other questions, I'd be happy. Yeah, go ahead, Klim.
Yeah, that sounded nice. I like that idea of making many similar cells, but I'm working with taste buds, and there are cells of 3 different types, and I'm not ready with idea what to be modeled when differences between them are small, and they should be modeled as equal cells, something like UTP. So sometimes when you talk about variable parameters of different cells, that was exactly I thought of, and that's nice.
Yeah,
I'm not the only one, and there is already
So this
do it.
particular case, if I show you the model for the synapses, oops. In this model for the synapses, it's these V0 and Vsyn are the parameters that you can adjust. This is the synapse resting potential. And if you adjust that, you can make the synapse be inhibitory or activatory. So then it could be, there are people have tabulated values for specific types of glutaminergic neurons and dopaminergic neurons and the different kinds of neurons. There are parameters, these tau R, tau D, and V0 and Vsyn. These are the basic parameters of these synapses and their tabulation. So you can use the base model that has these parameters and then just adjust the values on each unit. Yeah. Okay, I think I'm going to stop sharing.